Center for the Ethiopian Educational Information and Communication Technology presents Educational Satellite Television Programs.
Hello students, my name is Asrat. I am your biology teacher on this revised lesson. Today I'm going to deal with, explain about cell biology in our short revised lesson. Cell biology is a science that deals with cell, its structure and functions. And the science is said to be cytology. And I hope you will remember some of the very important pointers in the first beginning of the lesson. Under cell biology, you need to remember some points like the development of cell theory, the discovery of cell, and the science of microscopy along with it. And the other lesson that I am going to focus on today will be on types of cells. Of course, you should also remember how to measure the size of cell, and also how surface area affects cells. Okay, let's proceed to the cell types of cells. Basically, we have divided cells into two major groups, if you remember. So I need you to remember these very important pointers at the end of this lesson. One of them is you have to be able to appreciate the two basic types of cells. We call them prokaryotic cells and eukaryotic cells. Okay? I need you to remember those very important cells called prokaryotic and eukaryotic cells. Okay? And the other very important point I need you to remember is you should be able to give examples of these cells and also explain the structural differences between these cells. And at last, you need to also explain about or compare about these two cells. Okay? You should be able to mention the differences between them. So let's go to these very important types of cells. I need you to know there are three domains of life. If you remember, the three domains of life belong to the groups of the domains of life includes prokaryota, eukaryota, and there is another one in between, which we call archaeons. Okay? I need you to remember archaeons as well. So it started from prokaryotes. When you say prokaryotic cells, these are known to be the very oldest kind of cells existed on this planet. They are as old as 3.5 billion years old. So we consider them as the first cells evolved on this earth. So they are known to be our ancestors, evolutionary. Okay? After prokaryotic cells, it is believed to be archaeons, or we call them archaebacteria, that actually came up after, and from which eukaryotic cells evolved. But basically, under this lesson, we deal with these two major groups of cells. Prokaryotic cells, as you know, are very small in size, and they are very primitive, and they actually are made of a very small cytosol. We call it cytoplasm. And inside it, you can see that they have a DNA strand, which is circular DNA. And they have also ribosome, very small in size. And they have some extra cellular structures like flagella, by which they move. They also have plasmid, extra DNA to help these cells actually somehow develop themselves to resist some antibiotics and some other things. So we deal with these two basic kind of cells in detail. The first one, as I mentioned, are prokaryotic cells. This name clearly tells you that cells with the first kind of nucleus, a nucleus without a nuclear membrane. So they lack a true or a distinct nucleus. Second one, These cells are actually well known as bacteria, basically EU bacteria and cyanobacteria. These two groups of bacteria are named as the only prokaryotic cells existed so far. All right? So when you see these cells, you will see some very important structures. We actually call them, one, the outermost layer of this cell. We call it the cell wall. Okay? You might also see the other part just below the cell wall, which is called the cell membrane, which is common to all cells as well. You also see that there is extra plasmid DNA, which is used as well for other important characters that belongs to these specific bacteria. And you might see also some cilia, some flagella that help these microbes to move around as they actually function. The second kind of cells we see in here are going to be the eukaryotic cells. Eukaryotic cells, as you see, EU means true. Okay? So when you say eukaryotic cells, it means they are the true cells with a nucleus. Okay? So they have a true nucleus, a nucleus with a membrane bound. Okay? It is bounded by a nuclear membrane. And it has well organized compartmentalized cellular structures. You might see also a very large number of organelles that are found suspended in the cytoplasm, and they have their own membrane inside it. For example, you might see mitochondria, you might see chloroplast, you might see Golgi apparatus, you might see several kind of different organelles that are found in eukaryotic cells, but they are absent in most of prokaryotic cells. This is also another example of eukaryotic cell called a plant cell. In plant cells, you might see some other extra structure called cell wall, and you also see a very large central vacuole, except that they are the same to eukaryotic cells in case of animals. All right? So, just to compare these two cells, I need you to summarize them in this way. One of them is the prokaryotic cells are very primitive and they are very small in size. They range between 1 to 10 micrometer. That means they are the smallest cells alive. Okay? And you might see also eukaryotic cells. They are a bit bigger than them. They are advanced, and their size is also 10 to 100 micrometers in size, which means they are bigger, and they have also developed very important organelles. You see the second one, nucleus. Nucleus, in case of prokaryotic cells, is not bounded by a nuclear membrane, while in case of eukaryotes, you can see the nucleus is well enveloped with a nuclear membrane, just like a cell membrane. All right? So the other one is DNA molecule. The DNA molecule in case of these prokaryotic cells is found simply suspended in the cytoplasm. It's not compartmented or separated. This makes it very primitive in its structure, and it is not also associated with histone that actually makes it chromosome. So they have a circular DNA than that of eukaryotes, while the eukaryotes have a linear DNA, and they also have histone that actually coils the DNA on it and makes a chromosome so that it carries hereditary information properly. Mitochondria, Golgi apparatus, lysosomes, and other many organelles are absent in prokaryotic cells. So most of the function in prokaryotic cells is carried out by the cell membrane than the organelles. So this cell membrane has to be highly folded inside of it in order to separate the chemical reactions that happen in the cytoplasm. But in case of eukaryotic cells, you see that many organelles are present, even they have double membrane organelles. You can see chloroplast, ribosomes, and cell wall. I want you to remember this one. Cell wall in prokaryotic cells is always present, and it is basically made of a different kind of chemical. We call it peptidoglycan. I need you to underline this part, okay? We call it peptidoglycan, all right? But in case of plants, you already know that it is made of cellulose. Okay? The other lesson that we are going to go to is about parts of cells. This basically refers to the cellular structures called organelles. They are usually found suspended in the cytoplasm. So I need you to know that these organelles and their specific functions. Okay? One of the points that I need you to know is the importance of the cell membrane. I need you to know that also the composition, the arrangement of the lipid and protein in forming this cell membrane. Okay? And there are several models developed about the cell membrane. So I need you to know those models and compare them in which one is actually preferred recently, and some other parts of the cell and their function. So these are the main targets of our lesson under this topic. So whenever you see a cell first, what do you see, students? The very common outermost layer of animal cells, we find the cell membrane. This cell membrane is a very essential part of the cell to provide a very little mechanical strings using its cytoskeletons, mainly made of microfibrins and microfilaments. At the same time, this cell membrane helps cells to control what enters and leaves out of the cell. Cell membrane is neither permeable nor impermeable to everything. It allows some materials to move in and out of the cell through different mechanisms. It might allow some materials to move by simple diffusion, and the other one maybe by facilitated diffusion. Some materials could move by osmosis. Necessarily, in case of osmosis, it is only water that moves across the cell membrane. The cell also can move materials by active transport like endocytosis, exocytosis, and some others. So those are the very important pointers that the cell membrane helps. In addition, the cell membrane is a very important part of the cell that helps us in cell signaling. All right? Cell signaling is a means by which cells communicate to one another. You know, cells talk. One cell discusses some other materials with the other one. For example, hormones might be sent to one cell of your body. That hormone is basically identified by the method of cell signaling. In plants, auxins, the growth regulators, they are also, of course, identified by the cells through the method of cell signaling. So cell membrane communicates with the environment of the cell with different chemicals, with different drugs that we take when we are sick by the method of cell signaling. This is aided by the cell membrane. Cell membrane is also a barrier of the cell. It separates the cell from its environment so that any kind of reaction happening inside the cell is well separated from its environment. Okay? So, generally, the cell membrane helps us in controlling what enters and leaves out of the cell. It controls the organelles that are found inside. It holds them inside. Okay? And it acts as a receptor site for recognizing of hormones, enzymes, drugs, even sometimes viruses. They actually come into your cells through the cell membrane. Of course, it might not be its function. And the other one is to detect some neurochemicals, neurotransmitters. So those are the basic uses of the cell membrane. So how does the cell membrane seem to you? What does it look like to you? Many scientists have done a lot of scientific models. You know the difference between model and theory. And they have developed different kind of models that explains about the plasma membrane. It wasn't understood until the invention of electron microscopes. You know that. So it was because of the scanning electron microscope that understanding of cell membrane became possible in our studies. Until then, many scientists developed the theory, the model that explains the cell membrane. And of course, basically, we all agree that the cell membrane is made of phospholipid bilayer. It's like two layers of phospholipid, okay? They come together and they form this bilayer of phospholipid. You can see from this figure that this is the phosphate, the hydrophilic, and the fatty acid chains, the brown ones, are the hydrophobic parts. And the other phospholipid layer facing opposite to a side so that they can, of course, form the membrane. But let's see the models. How do they actually develop the models? These are the models developed. You might see that one in your book. I don't have to go to the history of cell membrane models. But the very well-known model of cell membrane is Davison and Daniel's model. This model explained that cell membrane is basically composed of phospholipid bilayer. No doubt about that. But they actually explained in a different way. So they were not fully accepted during that time. But they only used the first electron microscope to explain the scientific model. They have to be appreciated. Okay, they said that it is a kind of sandwich of protein model. Okay, let me show you that in the picture. Okay, let's see the picture. For example, in 1935, they have actually developed this kind of model, which the protein is found like a sandwich, you know, burger, right? There is a bread on it and there is a meat in the middle and some vegetables. Just like that, the bread is on the surface and the phospholipid layer is in between. But as more evidences and more evidences of science came in and microscopy developed, scientists could not, these two scientists especially, they could not explain how molecules move across the membrane. So they actually developed it somehow after 20 years, and they have developed just a protein-lined pore that actually helps us in transport of molecules across the membrane, which is not enough to clearly convince the other scientists. So a new model has to be developed. That a new model is the FLIUD MOSAIC model. This model clearly somehow explains how molecules move across the membrane. It clearly explains also the distribution of proteins. That is different from the sandwich model. The sandwich model doesn't give clear explanation about the movement of molecules. So these two guys, Singer and Nicholson, proposed a totally different idea. They agree with basically with a phospholipid layer, just like this, but the protein is not on top, like a sandwich. It is rather studded or spun in between the cell membrane. This is a protein. Look, the purple color ones are the proteins studded in between the membrane. Just like this is a bilayer. Let me put this protein here, okay? Like this. These proteins are studded. They span the cell membrane, one. They help in transportation of molecules across the membrane. So they have explained in a different way. So their model was purely accepted and became more popular than the DIFFUSION's model. This is the DIFFUSION's model. That one yet is a FLIUD MOSAIC model. Let's see the main features of this model. This might be just a model or a kind of structure of the cell membrane, what a cell membrane looks like when you see it under a scanning microscope. You have a phospholipid layer. There are peripheral proteins. There are also integral proteins. There are cytoskeletons. You see that there are channel pores through which ions can move. There is also cholesterol that is helping in the control of the fluidity of the cell membrane, because the FLIUD MOSAIC model kind of has a fluid nature. Okay? And there is also one very important thing that I need you to know. That one is the glycoproteins, the glycolipids. You need to tell the function of each of these molecules found in the cell membrane. They usually occur on your national exam. Okay? The hydrophilic and the hydrophobic parts located in different positions. The outer layer is hydrophilic and the inner sections are hydrophobic. The fatty acids face inside. They are the hydrophobic parts. There are very important proteins called integral proteins. That includes intrinsic and transmembrane proteins, under which you might find three important transporting proteins inside a cell. One of them is the channel protein. This channel, as you can see, has a tube through which ions can easily pass through either facilitated diffusion or through active transport. That ones are called as pumps. There is potassium pump, there is sodium pump, you know those ones. There is also carrier protein. The carrier proteins also transfer molecules by changing their shapes. Okay? You also see glycoproteins. These are very important. If you remember GP120, in case of the cell T4 cells, it is a very important recognizing molecule. You know that where it is. Anyway, glycoproteins are the ones that are basically made of very specific kind of carbohydrate and protein molecule that help in cell recognition. Let's see each of them in detail. Here you go. The very important part of the cell membrane is the phospholipid layer. Then the integral proteins contain intrinsic proteins and transmembrane proteins. And this membrane, actually these proteins, one could be channel protein. They are channels. They pass the molecule around through them through facilitated diffusion or active transport. Okay? And there are also carrier proteins. They move molecules across the cell membrane. We have also peripheral proteins. They are found mostly on the outside of the cell membrane, and they help in spanning the two membranes together. They hold them together, like a screw holds some kind of utensils in the house. Okay? So they might have a function of enzymes also. They might be used as hormones. They help also to hold on the cytoskeletons that I have told you they actually give mechanical strength to the cell membrane. Okay? Glycoproteins and glycolipids. This is I want you to focus very seriously, because glycoproteins are the ones that are used as signals to other cells. That's how our cells recognize other foreign antigens. So glycoproteins or glycolipids make antigens of the cell surface membrane. They help to recognize one cell from the other. They also act as receptors. Okay? Receptor sites for hormones, for drugs, for any kind of chemicals. You know that cholesterol is not important in our diet because we already produce it in our liver. And this cholesterol is a very important part of the cell membrane. If you see this cholesterol, it is used as a very important part of the cell membrane to decrease its fluidity. Okay? So, we might go to the next one then. Once we finished the cell membrane, how do these molecules pass across the membrane? Materials move across the membrane, as I mentioned, through a semi-permeable membrane. The cell membrane is like a traffic. It doesn't allow everything to move in and out. So it controls some things to pass. What kind of things pass by simple diffusion? Simple molecules, simple enough to pass through the membrane. 2, they have to be lipid soluble, nonpolar, and they have to be non-charged. Okay? So unless the cell membrane won't let any molecule to pass across the membrane. So how do they move? In 2 different ways, by passive or by active transport. Passive transport is a mechanism by which molecules move from higher concentration to lower concentration, just due to concentration gradient. This doesn't need any kind of ATP or energy expenditure. In case of active transport, cells move molecules from lower to higher concentration. This needs some kind of ATP or energy to be delivered to the cells. So for that, cells, of course, transport. What kind of molecules pass by active and passive transport? Okay, in case of simple diffusion, it could be carbon dioxide, just moving out of the cell to your body, to your cell environment, then out of your body. Oxygen by simple diffusion. Okay, and the other kind of simple diffusion is osmosis, that water moves from in and out of the cell. In osmosis, you might remember some things. Facilitated diffusion, it's a passive transport, but it is assisted by some channel proteins to allow molecules move in and out of the cell. It can't happen without these channel proteins. In case of active processes or active transport, you might remember endocytosis. Endocytosis, when a cell takes in some molecules into the cell, into the cytoplasm. It could be done by different mechanisms, but this also requires energy because as they take larger particles into the cell. And exocytosis, when a cell removes or excretes some things like enzymes, waste products, hormones, okay? So the cell has to do such kind of mechanisms in order to transport materials. So this is just a picture of how simple diffusion happens from high concentration to lower concentration until equilibrium is attained, all right? And in case of facilitated diffusion, you can see that. You see the channel protein? It moves molecules in and out through the channel. And in case of carrier proteins, the carrier proteins help in this transportation as well. But in case of active transport, when a molecule passes, there is ATP that should be hydrolyzed, and energy must be released for the transport of that molecule. other parts of the cell then. What do you see when you tour into the cytoplasm? Let's say you are touring in the cytoplasm. You first see the nucleus at the center, mostly in animal cells. The nucleus contains nuclear envelope, nucleolus, chromatins, or we call them chromosomes when they condense. This envelope helps us to move, control what enters and leaves out of the cell. The nucleolus is a center where the materials for synthesis of ribosome are prepared. You know ribosomes. Chromatin is a very important thread-like structure that carries hereditary informations in the cell. So the nucleus is supposed to be, or called to be, the central unit of a cell. Why? Is it because it is at the center of the cell? Yes, because it controls every activity of the cell. Okay? So the center, we call it even the central core. Okay? We go to the other part of the cell called mitochondria. You find this very unique structure of the cell in the cytoplasm. There could be thousands or tens of thousands, depending on the metabolic activity of the cells. This mitochondria and also nucleus, they both have double membrane, so they are called double membrane organelles. The mitochondria, as you can see inside, is very highly folded, called cristae. The fluid-filled part called matrix, and this part of the organelle is used for production of energy, so that we call it the powerhouse of the cell. And this mitochondria is the one that provides energy even when I am now talking. This ATP is produced in the mitochondria when glucose is broken down into its simpler forms. We'll come to that later. The other very important component of the cell is ribosomes, factories of proteins. Or we call them the sites, places where protein is synthesized. Of course, for protein synthesis, a nucleus must send information to through the messenger RNA, and that message that is sent through the messenger RNA must go to the ribosome to be sent the proteins required. And this ribosome has two components, the ribosomal RNA and the larger subunit, which we call the ribosome itself. We have also the other very important part, which works as a channel inside the cytoplasm, endoplasmic reticulum. As you can see from the figure, that it has several pathways through which molecules move inside a cell, through vehicles, if you believe. There are some vehicles inside that transport materials. And this endoplasmic reticulum is a membrane system. It is a channel through which materials are transported. And you might see two kinds of endoplasmic reticulums inside a cell. One is called the rough one, because it is attached with granular structures. Ribosomes are attached on it. And so that this endoplasmic reticulum produces protein, transports protein through vehicles to where they are necessary, maybe to Golgi bodies for modification, to their others. Smooth one is agranular endoplasmic reticulum. It doesn't have ribosomes attached or anything. So it just helps in the synthesis of lipids, steroids, and it helps in detoxification and metabolism of carbohydrates. So these two parties or components of the endoplasmic reticulum help in generally transportation. There are several other organelles called Golgi apparatus, or we call them Golgi body. This molecule helps in modification of proteins in general. Proteins synthesized by ribosomes brought to Golgi apparatus to be modified, packed, and concentrated. They will be concentrated, packed, and sent. It's like a post office. It actually receives the molecules, packs them, and sends them outside of the cell or inside the cell to mitochondria, to nucleus. It is a kind of delivery molecule. It packs and delivers, packs and delivers. So it is very important in such kind of functions. It makes basically glycoproteins, okay, as a sac. Lysosomes, we usually find them in animal cells and lower plants. They're usually called suicide bugs, if you remember, in lower grades. because they have a digestive enzyme that can even kill the cell itself. So you can understand simply from what they are, from what I said about their function. They are used to break down cellular debris, waste materials, so that these waste materials can be simpler and removed out of the cell. Especially the white blood cells carry a lot of lysosomes to kill bacteria, break them down, and remove them out of our body. Okay? And lysosomes are formed in Golgi bodies. You might not remember these ones, called centrioles. They are found at the top of the nucleus, and they help in cell division. If you remember the sister chromatids. The sister chromatids align themselves at the center of the cytoplasm. And when cell division starts, the sister chromatids or the homologous chromosomes separate. How? By the centrioles. They produce this spindle fiber and they pull them into opposite sides. So this helps. They usually become visible, of course, during cell division, but they help in cell division. Cell wall, you also find it in plant cells and bacteria, made of components called cellulose for plants, peptidoglycan for prokaryotic cells. Don't forget that. So it is completely permeable. It provides mechanical strength, maintains shape, controls turgidity of the cell from bursting out. In which animal cells burst? Because they have no cell wall. You remember that? Vacuole, very common, large, at the center, stores a large amount of cell solute called cell sap. It has a mixture of many salts, food, and waste products even. It helps plants, by the way. Vacuole helps plants because it is very important for their turgidity. It keeps them turgid so that leaves stay erect even they are thin during photosynthesis. So it maintains the osmotic balance of the cell. It gives them more turgidity, and that is important for plants. The last ones, but not the least ones, plastids. Okay, chloroplast, the other very important double membrane organelle in plants, of course. This is the most important organelle that makes you food that you eat right now. This chloroplast absorbs light energy, turns that light energy into food like glucose, lipids, proteins, so that food becomes available. And this is found in plant cells. This chloroplast contains green pigments in the grana of the chloroplast, and of course it makes the food. Sometimes you might also find chromoplast, but not in the leaf. This is common in the leaf cells, like palisade cells, but chromoplast is found in the flower, rather, in the fruits. Okay? Color of the tomato. Isn't it beautiful? It's because of the chromoplast. Leucoplast is not pigmented. So it's found somehow in the stem of plants. It's colorless plastid. So you have three plastids here. Okay, this chloroplast, as I have mentioned, is double membraned. Two regions. It has grana and stroma, where photosynthetic processes take place. We will see that in detail in the coming lesson, all right? That is how chloroplast does its function. Absorbs light, makes food, provides you the food. Of course, the extra food. They use it. And so organelles are, how do we know all these organelles anyway? All these, the ones that I have mentioned, are known through the mechanism of cell fractionation. Scientists separate these organelles using a machine called ultracentrifuge machine. This machine rotates at a different speed to separate the bigger ones like nucleus and the mitochondria, a little bit bigger one, lysosome, peroxisomes, it goes on until it separates the very smallest one called ribosomes. Okay? So those are the main points that I need you to remember in this lesson. Parts of a cell, types of cells, prokaryotic and eukaryotic, compare them, see their difference, okay? And I need you to know also the function of each organelle and the models of the cell membrane. Thank you. Have a good day.